as I explained the last time, the way we have filled this webinar series has been to go back to our poster presenters from the Inform meeting, with the assumption that most of you didn't really have a chance to go see those and chat. So we're starting with our winners, and we're trying to pair a senior person with a more junior person. And so today we have, hang on, let me get this in front of me so that I'm not staring off into space. Khalid Al-Tibi, who says that he got his bachelor's degree from Philadelphia University in Jordan. I didn't know that was a place, so now I do. He spent some time at Al-Khaldi Hospital and then moved to Freiburg in 2019, where he is now working on his PhD with Sarah Tucci. And I don't think I have the title of his talk in front of me, but he'll tell you what it is. He was our overall winner for the Inform poster judging. He was judged to have the best poster, and this was for everybody, so included all the senior people. And as a junior, as a PhD candidate, that's quite an accomplishment. So Khalid, we'll turn it over to you. You should be able to share your screen, and we'll looking forward to your talk.
Thank you, Julie. Thank you for the introduction. I'll just share the presentation.
Perfect. And if you put it in display mode, we'll be able to see it a little bigger. There you go.
Okay. Hello, everyone. First, I would like to thank you for giving me the opportunity to present our data here. My name is Khalid Al-Tibi, and I am a PhD student, and I would like to present our very interesting data on an inborn defect of the mitochondrial fatty acid biosynthesis. During my presentation, I will give you an introduction about the type of fatty acid synthesis. I will show you the link between mitochondrial fatty acid biosynthesis and lipoic acid biosynthesis, and the role of lipoic acid. Then I will explain the aim of my work, the study design, and the result that we got. Finally, I will finish with summary and conclusion. There's three types of fatty acid synthesis. The classical one is cytosolic, very well characterized, typical for mammals, and it consists of multifunctional complex. Also, it's responsible for synthesis of fatty acid up to C16 to C18. The type 2 is not well characterized, and it's found in archaea, bacteria, and a little bit in plants and some fungi. Every single protein catalyzes as a separate step, so they are single proteins. And what most of people don't know is the existence of the third phase in the mitochondria in mammalian cells. Sorry, it's responsible of the synthesis of fatty acid up to C8. The fatty acid biosynthesis pathway is directly connected to the lipoic acid biosynthesis pathway. It's started by activation of myelinate to myelinal-CoA by ACS3. The end product is the octanoic acid C8 used for biosynthesis of lipoic acid. The lipoic acid derived from C8 is a cofactor of all these mitochondrial enzymes involved in energy metabolism, like, for example, pyruvate dehydrogenase complex and alpha-ketoglutarate dehydrogenase involved in citric cycle, and also other important enzymes. So the mFAS2 pathway is not well characterized, but it's known that malonic acid inhibit the complex 2 of the mitochondrial respiratory chain, and that acyl ACP is essential for OXPHOS assembly. So mFAS2 play a role in the mitochondrial respiration. That's why the aim of our work is to evaluate the role of mFAS2 in the regulation of energy homeostasis. For our study, we used a human fibroblast from patient with a defect in ACS3 as a model or a tool for an for mFAS2 defect. P1 is patient that was diagnosed few years ago in our hospital, and P2 and P3 were a kind gift from Professor David Rosenplatz. As we know, ACS3 is an enzyme involved in converting melanoic acid into melanoic-CoA. A mutation in ACS3 increases the amount of melanoic acid. Concerning the ACS3 deficiency, the patient show an accumulation of melanoic and methylmalonic acid in urine considering the ACS3 mutation. This disease is unlikely, underdiagnosed, and considered sometimes as a non-disease, and has an incidence supposed to be about 1 to 30,000. Patients have very heterogeneous neurological phenotype. So the study design consists of checking the metabolic phenotype using Seahorse technology, the proteomics profile by Silac-based analysis, the lipolation degree by Western blot analysis, the role of amino acid in RNA reproduction, and finally, the lipidomic profile by lipidomics. First of all, to confirm the biochemical phenotype in ACS3 deficient, fibroblasts were cultivated, patient fibroblasts, and healthy control over different conditions. And we found that all ACS3 deficient fibroblasts show a methylmalonic acid and malonic acid accumulation, like in patient urine. Starting with the characterization of the metabolic profile, we use Seahorse technology and find out that the metabolic flexibility is disrupted. A blue represents the control, and red, green, and purple are the patients. Indeed, the mitochondrial respiration is affected. We see a decrease in the spare respiratory capacity with no difference in the ATP production. Then we checked the other energy pathways, and we found that the glycolytic flux was also reduced in the patient comparing with the controls. Finally, we checked the metabolic flexibility of the cells, and we realized that the patient cells show low flexibility and high dependency on fatty acid oxidation, suggesting that they need beta oxidation for energy production. To perform proteome profiling using Silac-based proteomics, 3 cell lines, including a control and 2 of the patient cells, were incubated with 3 different medium labeled with light, medium, and heavy isotopes, respectively. Then we prepared the pellet and the protein lysate, and we prepared the combination with ratio of 1 to 1 to 1 of these 3 lysate, and we sent it to the proteomic facility to do the analysis by tandem MS. And they sent us the result that we analyzed, and we got very interesting data. Around 1,600 proteins were identified. Out of these, around 200 were mitochondrial proteins. We clustered the common up and down regulated proteins in the 2 patient samples, and we found out that there is a fatty acid metabolism network commonly regulated, and we observed only few common downregulated protein. Because of Seahorse experiments showed a reduction of glycolytic flux, we evaluate the proteins involved in glycolysis that were detected in Silac. We can clearly see that the glycolysis was downregulated in both patients. So we assumed that this would be induced by a defect in lipolation of some enzymes. So we check the lipolation degree by Western blot with anti-lipoic acid, antibodies binding to different lipoylated proteins, and that quantification show a decrease in the lipolation degree of alpha ketoglycerate in the 3 patients and of beta pyruvate dehydrogenase in 2 patients. We have already observed a reduction of glycolytic flux and upregulation of beta oxidation. In order to evaluate whether the amino acid also contributes in energy production anaplerotically, we observed a significant reduction of some amino acid that feed anaplerotically the citric acid cycle, whereas some other amino acids were unaffected. In particular, the concentration of leucine, isoleucine, threonine, aspartate, and glutamine was significantly reduced in ACS3 cells, indicating that anaplerosis may compensate for hypofunctional MTFAS and meet the cellular energy needs. It's known that the MFAS2 is responsible for biosynthesis of fatty acid up to C8. And in yeast, the isolated enzyme were theoretically able to synthesize longer chain fatty acid. That's why we decided to check the role of MFAS2 in the cellular lipid homeostasis. And we performed lipidomic analysis. 1,420 lipid species were detected. The results show significant difference. What's interesting in the statistical analysis that we can see that there is a high concentration of ceramide phospholipids in patient fibroblasts, and that the cardiolipin, triglyceride, and sphingomyelin were increased. We also observed that twofold higher accumulation of the storage lipid triglyceride was accompanied by an altered profile of fatty acid chain length and the presence of odd chain species in ACS3 cells. The accumulation of cardiolipin was 2 to 4-fold higher for the cardiolipin of total chain length of 68 to 72 carbons. We also observed a significant increment of species with a higher level of fatty acid desaturation compared to healthy control. We observed also the exclusive presence of CL76 and CL72-8 in ACS3 called sorry. So we assumed that the reduction of respiration could be due to the shift in the compensation of CL, as they are essential for OXPHOS assembly. So to summarize, in our ACS3 defected patients, we have seen first an accumulation of molinic acid and probably a decrease in C8 ACP. So this is the reason of the decrease in the mitochondrial respiration. Also, a decrease in C8 ACP will induce a decrease in the lipolation of pyruvate dehydrogenase, so a decrease in glycolytic flux. But we see upregulation in beta oxidation pathway, and that could be a compensatory system at that level. From the other hand, we also assumed that the decrease of C8 is the reason of the alteration of lipid profile seen by lipidomic analysis. So to conclude, we proved that the MFAS2 plays a role in an energy homeostasis and in metabolic flexibility. And it's not only responsible for the biosynthesis of fatty acid C8, but also for lipid homeostasis of the whole cell. I would like to thank my supervisor, Dr. Saratucci, and our former colleague, Zainab Febin. and everyone who contributed to this work, and you for your attention.
Great, thank you very much. We have, I failed to mention that we asked our speakers to try to leave plenty of time for conversation. So please feel free to unmute yourself and ask questions verbally. If you don't feel comfortable doing that, go ahead, type them in the chat, and I'll take care of announcing them.
Very nice and very interesting work.
Thank you.
My question is that obviously malonic acid effect on fatty acid oxidation on one side increase all of the activity of the enzyme, but on the other hand, what happens when you actually measure fatty acid oxidation in this cell? So malonic acid could also have an effect on CPT1 and actually decrease this effect. Did you measure the actual fatty acid oxidation in these cells?
Sorry, what's the last part?
The question is whether you measure the actual oxidation of fatty acid in the fibroblast of patient with combined methylmalonic acidemia.
Yeah, I think that's really good idea, but we did not do it.
Not yet.
Not yet.
Thank you.
Khalid, with all of the changes that you're seeing here, why aren't these patients more symptomatic? This is such a strange biochemical phenotype that looking at some of what you're seeing compared to some of what we're seeing, for example, in the bona fide long chain fatty acid oxidation defects, they're equally large. Why are these patients so mild to the point that everybody's willing to say that this is not a disease?
I think because the heterogenic phenotype, I think it's because some people are considered like not disease, right? So
May I add also something? Unfortunately,
Yes, please.
it does not work. But we are working together with a group from Finland. And this is the group of Alexander Castagnotis, and he characterized this pathway very well in yeast. And actually, there is some unpublished work from his group. And there is another enzyme in the mitochondrial membrane. This is localized in mitochondrial membrane, which is able to cover in part the activity of ACSF3. So maybe one reason could be that, of course, not all mutation shows the same severity in all the patients. And the second point is how much this second enzyme is really able to cover for the activity. So that maybe, yeah, this could be in effect, of course.
Mm-hmm. Yeah, I can. Let's see. We have Barbara Bacher. Yes, please step in and ask.
Thank you very much, Khaled. That was really interesting. I was wondering, can you explain why glycolysis is going down? How would this be regulated?
I would say, like I said in the why the you are saying, like you want to ask why the glycolysis is affected, right?
Yeah, right. How could this be regulated? I mean, I see that you measured a few of the enzymes, that their protein is down.
Yeah.
But what would be the signal for this?
Because as we know, like the C8 ACP is down, so it should be the lipulation is down. So we can say like the lipulated like pyruvate dehydrogenase and alpha ketoglutarate also like going down. So this is could like decrease the glycolytic flux. I would say that.
But I thought you were talking about lipoylation of the mitochondrial proteins.
Yeah, but maybe like it's as a compensation, like going toward the beta oxidation, and that's block the going to glycolysis, like
Thank you.
taking another direction.
Thank you.
So Zara, go ahead and dive in. Please explain your thought from the chat.
Thank you. So we did not measure this yet, but I can imagine that this could be also because of an alteration of redox equivalence. I don't know exactly at which level, but if we have an impairment in the activity of pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase, maybe there is some kind of feedback mechanism and alteration of at this level that inhibits somehow the glycolysis outside the mitochondria. We don't know yet exactly how this works, but this is at the moment just an idea why. Because inside...
But isn't it often the opposite then, that if you have a mitochondrial defect that you would produce more lactate, have a higher glycolysis via similar mechanisms as what you're...
Yeah, this is true. To be honest, I don't know. We didn't check yet. This was really quite surprisingly for us.
Yes, I fully agree, and really intriguing.
Yes.
Do you see an increase in reactive oxygen species in these cells?
We did not measure this yet.
Oh, we're giving you lots of good ideas.
Yeah.
Another question about the cardiolipins. We spend a lot of time worrying about cardiolipin deficiency in Barth syndrome, and we're seeing it in the trifunctional protein and LCHAD deficient patients, as we talked about last month on this webinar. What's the implication of increased cardiolipin, and especially I'm wondering, is this somehow or other affecting your respiratory chain, or is that are those two unrelated?
Should I answer, or Khaled, do you want to answer?
Khaled, you can try, and then Sarah can bail you out.
So as we know, the cardiolipin, I think, increase the make upregulation for complex 4, if I'm not wrong. And I think that's why they are onboarded in here.
Yes, actually, the right combination or the right cardiolipin profile associated with the desaturation degree and maturated cardiolipins are essential for the assembly of OXPHOS. So if we have a deregulation in the cardiolipin structure, maybe we also have a problem in the assembly of OXPHOS, in particular of complex 4, and therefore in the electron transfer.
Have you looked at mitochondrial morphology in these cells, or super complex species in the cell lines?
No, not yet, but I have to say we have a full list of things that we want to do and we are planning to do, but the poor Khaled is working on 3 different projects, and sometimes it's quite difficult to coordinate all 3 of them. So yeah, we need people to do the experimental work, of course.
Yeah.
Yes.
But Zara, I still come back to the question, with all of these cellular derangements, why are the patients so mild? Any thoughts on that?
No, not really. And I am not sure that the patients are really mild. I think the disease is underdiagnosed, because maybe it could be that in several cases malonic acid is not detected. But I can imagine that maybe some patients with higher methylmalonic accumulation, methylmalonic acid accumulation, could be actually ACSF3 deficient patients. If the gene is not sequenced, you can never exclude that that they are not ACF3 deficient.
Christoph Olendahl has a question. Go ahead, Christoph.
Yes, thank you very much. Thanks, Khaled. It's a really good presentation. I was wondering if you had any idea of the activity of this mitochondrial Fas enzyme. How does it compare to the activity of the cytosolic of its cytosolic cognates? Is it a much lower activity or comparable to what you see with the cytosolic synthases?
The cytosolic synthase, it's increasing. The idea that I didn't study the cytosolic MFAS, I was focusing in the MFAS 2, so I don't have like that much of information about that.
Okay. Do you have an idea of?
If I may answer, the activity is very similar. Actually, it is described in human mitochondrial that in the mitochondrial fatty acid biosynthesis, the synthesis stops when the chain length reach C8, so octanoyl ACP fatty acid. But actually, if you express, if you do overexpression of the every single enzyme of the mitochondrial fatty acid biosynthesis, you can actually synthesize fatty acids up to C18, C16, like normal fatty acid, like the normal cytosolic fatty acid synthesis.
Oh, okay, very interesting. Thank you.
You're welcome.
And Grant Hatch comments in the chat that he suggests looking at increased cardiolipin synthesis as a compensatory mechanism.
Mm-hmm. Yes.
Grant, what would that look like? I mean, what do you think? What are you thinking there?
So I'm thinking along the lines of that might be potential compensation for increased oxidation, but because you have changes in the fatty acid composition, that results in the bioenergetic deficiency, right? So I think, I mean, that's why I think it might be interesting to look at the synthesis. But also one of the things that I noted in your slides is that, and correct me if I'm wrong, but phosphatidylethanolamine was not changed, was it? Its mass, at least from yeah. So phosphatidylethanolamine is a major mitochondrial lipid as well, and I'm just wondering if might be potentially a turnover defect going on, which accounts for the accumulation of some of these lipids in the mitochondria. Just as a side thought. I mean, these are another rabbit hole you can go down, but I mean, it's an interesting observation. So, yeah. Excellent talk, by the way. Thank you very much.
Thank you for your suggestion.
Very informative. So.
All right. Thank you, Khalid, for that great talk, and everybody for the interesting discussion. And we're going to go ahead and do our second talk. This is from by Yudong Wang, who is an assistant research professor here at the University of Pittsburgh, and works in my group. His poster was selected as the top poster for senior investigators at the last Inform, and he's going to go ahead and present his studies on the continuing to explore the interactions of long-chain fatty acid oxidation in the respiratory chain. Yudong, go ahead and share your screen and start.
Yeah, okay. Thank you for the introduction, Jerry. And hello, everyone. My name is Yudong, and I will presenting our Inform 2020 poster today. So the title is defining the structural and the functional role of the VRCAD in the FAO and the ETC supercomplex, or MSC, macromolecular supercomplex, through the recombination with the VRCAD to the VRCAD knockout mouse heart mitochondria. Yeah. So I would like to talk about the background related to this project. So in our recent publication, we established existence of the macromolecular supercomplex, it's MSC, due to the following three protein interactions within the fatty acid and electron transfer chain. So first is the TLP interact with the NADH binding domain of the complex 1 of the electron transfer chain. So second interaction is between the ETF dehydrogenase and the complex 3. The third is the VRCAD physically interact with the trifunctional protein. So this MSC structure allow the fatty acid oxidation can safely transfer electron equivalent NADH and QH2 to the electron transfer chain and finally generate ATP. So this project focusing on the further investigating the functional role of the VRCAD in the MSC. So my hypothesis is that because of the existence of the MSC is true and that VRCAD plays an important role in the MSC. So then if we knock out the VRCAD, it will significantly damage the MSC structure, and that's going to affect the flux from the fatty acid oxidation to electron transfer chain, and also going to generate increasing the RS generation. So reversely, if we recombine the VRCAD into the VRCAD knockout mitochondria, and that will restore the structure of the MSC, and thereby improving the flux from the FAO to ETC, and decrease the RS generation. So through this research, the functionality of the MSC can be confirmed, and the role of the VRCAD can be investigated. So now, how did we plan to test this for this hypothesis? So, to test our hypothesis, we are the mitochondria isolated from wild type and VRCAD knockout mouse heart. And recombination was achieved by the incubation VRCAD knockout mitochondria with the purified VRCAD protein. So the following method was used, and first is the blue native gel electrophoresis to see the change in the supercomplex. The second is the two-dimensional SDS page Western to see the individual protein changing in the supercomplex. The SDS page Western going to test the total amount of the individual protein in the mitochondria. So this first one is the flask assay for the from the fatty acid oxidation to electron transfer chain. The last one is the RS generation assay. So I will first discuss our blue native gel electrophoresis result. Okay. So we can see on the left side is the blue native gel analyze the wild type, VRCAD knockout, and the VRCAD recombination sample. So, and we can see that the knockout, the medium line, the knockout of the VRCAD caused the around 50% decrease of the supercomplex. However, once we recombine the VRCAD into the mitochondria, we can see that the almost 90% of the supercomplex on top is recovered. So this result was supported by the complex 1 activity stain, which is on the right side of the. So we can see the complex 1 activity also dropping down in the knockout VRCAD. and the VRCAD recombination restore most of the activity of the complex one. So in addition, we can see in the left GR, we can see that the complex 5, 3, and 4, and 1 also, we can see slightly reduced in the knockout and restored in the VRCAD recombination. So next, the complex 5 activity GR-STAN, and that we can see that in the VRCAD knockout on the right side of the GR, we can see the monomer almost 50% disappear and the dimer completely lost. And in the VRCAD recombination, we can see that the monomer completely restored and the dimer partially recovered. So next, we went to see what happened in the 2D SDS page based on the Blue Native. So this lists how the antibodies we used for this experiment. The top group is from the fatty acid oxidation, and the second is from the complex 1 NADH binding domain proteins, which is NDUFV2 and NDUFS1. So through the Western plot, so we can see that we horizontally put the first dimension of Blue Native on the SDS page here. So we can see the first GR, so VRCAD knockout, so VRCAD not appear in the entire GR. So, however, in the recombination of the VRCAD, we can see on the blue line on the left side is the supercomplex. We can see the VRCAD automatically appear in the supercomplex part. And also we can see the TLP alpha and the beta dramatically reduced in the VRCAD knockout. So however, it's reduced in the recombination with the VRCAD. So the third one is the ETF alpha. So we can see that the VRCAD knockout, the ETF alpha no longer appear in the supercomplex. However, in the recombination, we can see the ETF alpha appear again in the supercomplex. So the first one is the NDUFV2, I'm sorry, which is the NADH binding domain of the protein. So we can see that VRCAD knockout largely reduced the amount of that protein in the supercomplex. However, the VRCAD recombination can significantly restore that protein in the supercomplex. So the protein present here shows no change in the VRCAD knockout mass. So we can see that ETFDH and CPT2, RCAD, MCAD, SCAD, and the last one is NDUFS1, which also are the NADH binding domain protein from complex 1. So this suggests that not all the proteins in the supercomplex are affected by the knockout of VRCAD. So as a control, the purified SCAD was incubated with the isolated hard mitochondria from the VRCAD knockout. So result showed that that recombination do not affect the content of SCAD and the VRCAD in the knockout, and even in the combination of the SCAD. So this further provided the evidence support the VRCAD play a role in the formation of the MSC, and is accompanied by certain FAO and ETC proteins. So next experiment, we would like to talk about the SDS page where the Western test to see the individual protein what happened. So in this, from on the left side is the FAO proteins. And there's 3 columns. We can see the wild type, VRK knockout, and the right side is there is the compensation with the VRK. So we can see on the right side is the ETC proteins. So we can see most of them are not affected by the knockout of the VRK. So only the exemption is the ETF alpha. Looks like in the knockout reduced 50%, and the VRK recombination do not change that total amount. So the red marked is the change we can see in the 2D Western, the above one. So this result tells us that not most of the total protein level did not change in the VRK knockout. So therefore, the changes we observed in the supercomplex in 2D gel should be attributed to the presence of the VRK, where the knockout of the VRK negatively or destroyed and affecting the MSC structure. So this further suggests that the recombination of VRK result in the reassemble of the MSC. So next is, and we want to talk about the FAO ETC coupled flux assay. So this assay observed the flux by measuring the reduction of the cytochrome C and in the presence of the FAO long chain substrate. For instance, the C16-CoA, the C20-CoA, and the cytochrome C is the protein component in within the electron transfer chain. So this is the assay result. So we can see that the green one is the knockout, VRK knockout. So this flux activity dropping down around only 20% compared with the wild type, and not affected that much for the C8-CoA. And once we recombine with the VRK, so we can see the flux activity get recovered almost to 90% compared with the wild type. So as a control, we test the recombination of with the SCAD. We can see the 16-CoA do not change at all. So these findings suggest that MSC flux can be improved by the addition of the VRK. So the last one, we used the RS generation assay to observe whether VRK recombination is able to decrease the RS generation. So this assay revealed that we can see that the green one is represent the VRK knockout RS level. So it's 30% or 50% more than the wild type. And once we recombined with the purified VRK, so it was to be able to reduce the RS generation. Compared with the knockout, we can see the 20 to 40% of the RS get reduced. So this result suggests that the VRK also are necessary component for the MSC function. Based on our finding, so we would like to as we already hypothesized that once we the knockout VRK, the mitochondria recombined with the purified VRCAD. So we can see that is VRCAD automatically reassemble into the MSC. So the flux interrupted by the knockout VRCAD is significantly restored in the recombination of the VRCAD. So RS generation is decreased. So we would like to conclude that the macromolecular supercomplex MSC can effectively channel and catalyze the fatty acid oxidation substrates. So VRCAD is a basic component of the MSC, and it can stabilize the MSC structure. The next one is the fatty acid oxidation and the electron transfer chain functionally and structurally closely interact with each other in the MSC. So this research provides likely implication for the diagnosis of fatty acid oxidation disorders. So here is my acknowledgement. Here is my the references. Thank you so much for attending, and I'm happy to take any questions.
Yudong, that was terrific. If anybody has any questions, please go ahead and unmute yourself and ask. While we're waiting for that, Keith notes in the chat that both presentations will be posted online at the bottom of the homepage, so you'll be able to look at these again, or if anybody asks you about them, you can get them.
So Yudong, can I ask a question, Jerry?
Yes. Yep.
Yeah. So really nice talk. I enjoyed that a lot, quite a bit. I'm just wondering, do you think that the protein itself, the mutation, would it be necessarily something that affects its interaction with the complexes, or would it be the mutation that impacts on fatty acid oxidation, which is more important, or are they equally important, do you think, in terms of controlling how it works?
Yeah, oh, that is a nice question. So because the normal purified VRCAD are able to recombine into the supercomplex, so we already have the mutation purified VRCAD protein also, so we can do that recombination and the mutated protein VRCAD into the knockout. So by that way, we are able to just test and the specific case of the VRCAD mutation to see how it is changing for that kind of mimic the patient situation.
Yeah, Grant, keep in mind that these animals are null; they're deleted. So
right.
we're talking about no protein versus normal protein.
Right. Yeah. Okay. Thank you. Good.
Pablo, you have a question? Please go ahead and ask.
Yeah. Thank you, Jerry. Terrific talk, Yudong. My question is, I'm curious about the mechanism of ROS generation when the flux is decreased. Do you have any hint about that?
Yeah, the RS generation normally is caused by the leaking of the electron where is the transfer, and because of the mutation and the interaction of the protein itself, the structure changing, so that causes the leaking. So because the VRCAD not there, so the long chain fatty acid CoA form cannot completely smoothly oxidize, only could be oxidized by the partner of the ARCAD and ACD9 or some other proteins. So however, that not smoothly, so that's going to generate a large amount of RS can leaking out, generate RS. So that is a hypothesis.
And as Yudong alluded to it in his last slide, this really could have significant impact on or implications for treatment. You know, one of the things that's resistant to substrate alteration therapy, triheptanoin, is rhabdomyolysis, and so it's one of the things that we've been focusing on here is the possibility that the rhabdo is in essence not purely energy related, but related to the ROS that might be accumulating in muscle. Barbara, go ahead. You have a question.
Thank you very much. That was a really nice talk. I was wondering if this is an important component of the supercomplexes, would it also affect the oxidation of other non-fatty acid substrates? Have you investigated that?
Oh, we haven't done that yet, so sorry.
But you have shown, Yudong, that some of the other mitochondrial dehydrogenases similarly interact with the supercomplexes.
Yeah, for instance, the ARCAD, MCAD, probably also SCAD, only small the part interact with the supercomplex. So that is the next step for the research. So how those small amounts stay there, and the most part of the those proteins, dehydrogens, not binding to the supercomplex. But on the 2D GR, you can see that those, I guess, mostly appear in the medium part of the GR. That is the interaction between this protein and in the S chart protein. Probably the molecular weight analysis looks like they are interacting with those proteins, just like not a TLP, but the S chart. Okay. So that is our next step to see, and those interaction proteins which are not involved in the supercomplex, but a smaller molecular protein, and probably that is the kinetic question is, if we require those guys going to link with the supercomplex, all these like to stay in the matrix, because the catalyze, the substrate is hydrophilic. So there's a reasonable to think about those guys stay in the matrix. So how they are working, we can see in the model, we can see that the moving actually is ETF carrying the electron moving between the protein and from the supercomplex to the matrix proteins. So, and that is our next project. I think that, yeah, we need to further to work on it.
Could you speculate then whether the composition of the supercomplexes depends on the nutrient status? So for instance, if there are more fatty acid substrates, then it would be more, let's say the fatty acid pathway would be more integrated in the supercomplexes, and in another condition, it would be other enzymes, for instance, the pyruvate.
So, yeah, currently, when the reason is the interaction, because once the long chain coliform getting into the mitochondria, that is hydrophobic, and have to stay within the membrane. So that is why the VRCAD stay in the membrane and TLP stay in the membrane. So that is good for the transport of the hydrophobic substrate. So that is the why those VRCAD and ACD9 stay in the membrane. So that mechanism kind of a different compared with the membrane protein and the matrix protein. So the relationship we needed to further an experiment to see what happened. Also the blue native GR, we have to use the Daytonian detergent, which is very the mild of the detergent. So in the presence of the detergent, the they are able to forming we can see on the GR, we can see the supercomplex. So far, people think about there's 4 forms of the supercomplex with the different combination of complex 1, 3, and 4, even the complex 2. So that is the mechanism looks like very complicated. So we have to think about how we can accomplish this part of the research. So without the detergent, we had to run the Blue-native GR. So I tried. Currently, people using the 1 protein to 10 the deletoning, the ratio like that. I am dropping down that ratio, so that's going to save the interaction between fatty acid oxidation and the electron transfer chain. However, lower than that, so nobody published. We already done something on unpublished data. So, and that's going to cause more complicated supercomplex, even 10 band of the supercomplex appear on the GR. So that makes the situation more complicated. So, and we just thinking about recently without the detergent, what we can see about what the structure of the supercomplex. So that is a big challenge for us.
Barbara, that's a really interesting question, because we're currently viewing this as a very dynamic complex that they're not embedded in the membrane, so they can come and go to some extent, and the nutrient milieu would be really fascinating to study in that regard. I mean, that's a great idea. We'll have to see if we can figure out how to do that. I like that. I like that idea a lot.
Yeah, and it relates a lot to the work that Christoph Odendaal also will present in the next meeting, because what we observe is that the fatty acid oxidation is extremely strongly inhibited by its products. So if you could rapidly channel those products into a supercomplex like the NADH, FADH, that would really help. But I can imagine if you're not oxidizing fat, then you would not like to block the entry into the respiration for other substrates. So that is my idea behind this.
Yeah, that's great. Well, I hate to do this, but it's 11 and we have to stop. So I thank you all for attending, and terrific presentations and conversation. And I hope we'll see you all again next month.